Part 3 of emotional behaviors will discuss the attack and escape behaviors. Attack and escape behaviors are very related. If we think about the autonomic nervous system response, and we have this kind of fight or flight response, that's attack, fight, or escape, flight. We actually talk about it as a fight, flight, or freeze response. Sometimes we just freeze. He gives us the example of a cat playing with a mouse or other small animals. So we think of the cat, we watch this and it looks like the cat is playing and being really sadistic, right? They'll bat the thing around, look like they're playing with it, run off for a second like they're playing some game. But really what's happening is they're attacking, and then the autonomic nervous system is just active. And so sometimes they're reading that as fear, and they do the flight off, and they look and make sure it's safe, and then they attack again. So they're really dealing with that fight or flight and going back and forth. And in fact, if we give the cat a small enough animal, the cat will just go in for a quick kill. If we give the cat some sedation, the cat will go in for a quick kill because they're not having as much fear, so they're not doing the escape piece. If the animal is large enough, the cat just will not attack at all and be more likely to go on the escape route of that. So one of the reasons I really like to talk about attack and escape behaviors is so that we are talking about these different nuclei and these different areas of the amygdala, which I feel pretty remiss that we often miss the motivated behaviors. And talking about these little nuclei and these little specific areas of the hypothalamus, we did a little bit of that with sleep and wake, but they're these quite small structures in the brain. We've done enough research that we know quite a bit about the various nuclei and what they do. So the first one he starts talking about is the corticomedial area of the amygdala. And he talks about with this how we are building up activity there after we have attacked, and the fact that we have a higher probability of a second attack. So he gives us the example of hamsters. So if we see a hamster and then the second hamster comes in its territory, that first hamster is going to attack, most likely. And then we see if another hamster comes in his territory within the next 30 minutes, that hamster is even more ready to attack. We see even a higher probability of an attack. And that during that 30 minutes after the first attack, we're seeing this buildup of activity in the corticomedial area of the amygdala. So again, we're talking about this really quite small structure, as amygdala means something like almond, and it is this really small almond-shaped structure, bilateral. So we have amygdalae, as I sometimes say the plural. And you can see what he's done is taken this little piece and show us the magnification there. And we're going to talk about most of these areas, at least some of these areas. So we just talked about the corticomedial nucleus, where we see the buildup of activity after we have an attack and are likely to attack again. We are also going to talk, I'm pretty sure if I remember correctly, we're definitely going to talk about the central nucleus and the lateral nucleus. And I can't remember, sometimes there's a piece that I skip and it might be the basal nucleus. But there are some pictures of the amygdala and the kind of taking apart of this small structure and looking at those specific nuclei. As with many of our personality traits, our characteristics, we see effects of heredity and environment on whether or not we're likely to show violent behavior or to be violent. Some examples of the heredity aspect of this. So monozygotic twins resemble each other much more than dizygotic twins with regard to violent and criminal behavior. We also see that we tend to resemble our biological parents more so than our adopted parents in those adoption studies. We also see effects of the environment, though, as we know that if we are growing up in a household with violence, so if we are the victim or a witness of violence in childhood, we're more likely to show violent tendencies. Living in a violent neighborhood is associated with more likely to have violent tendencies, and exposure to lead. So a really different kind of environmental factor that's important to keep in mind is the potential for brain damage, and lead does damage an area of the brain, so really part of the prefrontal cortex that is going to make it more likely that a person becomes violent. As with many of our personality traits, our characteristics that are really more complicated, there is no specific gene that we can point to say we're going to be more likely to be a violent person or to have a lot of violent behavior. And as I said before, we're going to see an interaction between heredity and environment. One of the people who've looked into these interactions quite a bit is Avshalom Caspi. I think I say his first name wrong, but he's done a great deal of research. Many times he's done these longitudinal studies, and he has looked at specific genes and then interactions with specific environments. And so in this figure to the right, he's showing childhood maltreatment on the x-axis, so whether there was none and whether it was probable or whether it was severe. And then on the y-axis, they're giving a pretty complicated this composite score put together of antisocial behavior for these young people. And then they also looked at the activity, the genetics for the activity of the MAO, of that monoamine oxidase A. And so people who had genes where they had lower activity, if you remember, the monoamine oxidase is the enzyme that is breaking down all of those monoamines. They are taken back up in the process of reuptake into the presynaptic neuron, and then the MAO comes in and breaks that down, so we don't have an accumulation of those monoamines. So that's dopamine, norepinephrine, epinephrine, serotonin. So people who have low MAO activity, or the genetics for low MAO activity, we can see that they look just like people who have high MAO activity if there's no violence in their childhood, and even if there's probable violence. So what you can see is there's a crossover, but those are not significantly different from each other. It is when we see in their childhood severe maltreatment that we see a difference in these people, where people with high MAO activity are less likely to show antisocial or violent behavior, and people with low MAO activity are more likely. And this has been, some of his studies have not been replicated, but this one has been replicated several times. If we look in the animal kingdom, most aggressive behavior is committed by males. Okay, males competing for dominance, for females for territory. We also do see aggressive behavior in females when they are defending their young, specifically at that time. Male aggressive behavior heavily depends on testosterone, and in fact, I'm going to suggest that female behavior also, we're more likely when we have more testosterone in our system to show aggressive behavior. And I'm going to talk about this relationship with testosterone for a little while, and then I'm going to throw the monkey wrench in and say it's more complicated than that. But we're going to start looking at, or Calit starts by looking at some correlations with people who are in prison. So if we look at the people who have committed violent crimes, all right, rape or murder, what we see is they're more likely to have higher testosterone. So the people who are the red parts of those figures. And as we look at the less violent crimes, that are burglary and armed robbery, we still see this, they're likely to have intermediate to higher testosterone levels. And if we look at the people in prison who are there for drug offenses, that's clearly not a violent crime or an aggressive crime. They are the ones who have the lowest testosterone levels of the people in prison. Again, this is correlational study. So I am putting in this extra slide, which is repeating, and I'm hoping this flows okay with the PowerPoint, but I didn't want to have to redo all of that description. So I'm saying again that this is correlational, and I want to make it really clear that correlations are looking at associations or relationships. And so the causality, we don't know if A is causing B, B is causing A, or if there is some third variable. But I will go through this example of young boys who have aggressive tendencies, who can be impulsively aggressive. They go through, in one research study, they went through a training of just learning how to calm themselves down and do these breathing exercises and thinking things through in reaction to these aggressive impulses. And what they found was that lowered the likelihood of these testosterone surges. So it actually looks like there's a surge of testosterone a lot of times before having some aggressive behavior. But again, the directionality of all of this is unclear. And so we're going to talk about experimental studies. Researchers have experimentally increased testosterone levels in women. So women are typically low in testosterone. There are times during our menstrual cycle where it's slightly higher, but women are typically low in testosterone. And so they can experimentally increase this and then compare women with high testosterone to the more regular low testosterone. Terberg et al. Did this and found that women who received testosterone spent more time looking at angry faces. Wright et al. Did this, and they had women make judgments about visual stimuli, and the women did this either individually or in pairs. And then half of those women in each of those conditions were given testosterone, and then half were not given testosterone. When it came to making those judgments individually, the women did not differ whether they were higher in testosterone or not. But women who were making those judgments in pairs were less accurate. They were more likely to argue, and the dominant woman was the one who was more likely to make the decision, and they were not necessarily the person who was correct. This goes along with the research that shows that committees work more harmoniously if they have a high percentage of women on them. As I said when I started talking about testosterone, it is not that simple where we see just high levels of testosterone mean that I'm going to be more violent or have more aggressive behavior. They have also looked at serotonin. So lower levels of serotonin appear to be correlated with more aggressive behavior, more violent behavior. I'm going to start with some animal studies and then talk about a human study. So with the animal studies, I need to introduce this estimate of serotonin level or serotonin turnover, which they get as from a measure of 5-hydroxyindolacetic acid. I'm going to start saying 5-HIAA because it's even though it's a little bit longer, it's actually more simple to say. So this is serotonin's main metabolite that they can get a measure from the cerebrospinal fluid. So Valzelli has done several studies looking at male mice, and he often does this thing where he isolates them for some amount of time. So in this study, he isolated male mice for 4 weeks, and that increased their aggressive behavior. It also decreased their serotonin turnover. They also had strains of mice with just lower serotonin turnover, and they were even more likely to show aggressive behavior. They've also done a really fascinating study where they just examined monkeys after getting this measure of 5-HIAA, and they put those monkeys into categories of the lowest, the middle levels of 5-HIAA and the highest levels of 5-HIAA. So the monkeys with the lowest levels of 5-HIAA, right, they have the lowest levels of serotonin. And what they found was those monkeys were more likely to attack a larger monkey. Okay, and if you think about how much sense that makes, they're not likely to win that fight, and yet they're still likely to attack. And there were other measures of this, not just violent or aggressive behavior, but also impulsive behavior. So these monkeys with the lowest levels of 5-HIAA were also more likely to swing from tree to tree and make longer jumps, that they were less likely to grab on to the limb of the other tree. And few of those monkeys in the lowest category survived past the age of 6. In human studies, low serotonin turnover has been linked to people with a history of violent behavior and violent crime. They are more likely to have lower serotonin turnover. People who attempt suicide by violent means are more likely to have lower serotonin turnover, and there he gives us the figure of the levels of 5-HIAA in the cerebrospinal fluid, and that it is higher in those who've never attempted suicide and lower in people who have attempted, and then the lowest in people who have attempted more than once. We also see for people who get out of prison, those with lower serotonin turnover are more likely to go back to prison and show these recurrent violent behaviors. They find this relationship, this negative correlation with violent behavior. Some of these have not been replicated. It appears to be a slight correlation, and some of the replications, what they've tried to do is to look at answers to questions, and there doesn't seem to be any influence there. So it's really the aggressive and violent behavior itself. We can have changes in our diet that can alter our serotonin synthesis. And so for people who have some depression, I will probably end up talking about my depression more and more as we get into psychological disorders. But I know that for me, even just having a low tryptophan diet or a diet that's higher in phenylalanine can cause some depression. So I'm very careful with my diet. So tryptophan, if you go back to our biosynthesis, is one of the the amino acid precursor for serotonin. And then phenylalanine is the amino acid, or tyrosine phenylalanine is the amino acid precursor for dopamine. And so dopamine and serotonin are in the brain, right? They're not going to cross the blood-brain barrier. What's going to cross the blood-brain barrier are these amino acids, and they are competing actually to get across the blood-brain barrier. And phenylalanine and some of these larger amino acids that I've often heard of it talked about as they kind of bully their way across, and they're more likely to get across the blood-brain barrier than tryptophan. So even I'm going to include this because I like this study a lot, where they gave those, and I know I've told this story before in this class. I'm pretty sure I told this early in the semester, where they gave college males a diet high in aspartame, so sweet and low kind of sweetener, fake sweetener, and maize, so American corn. And those college males were more likely to get into violent altercations with each other than people who were not given this particular diet. So they're really raising their phenylalanine levels, which means less tryptophan getting across the blood-brain barrier, and they were more likely to have this kind of impulsive fighting. I have said, and I usually say at this point, because I love corn, and I do the little Charlton Heston of "you can take my corn out of my cold dead hand." I love corn, and I know it's bad for me, and it's one of my real weaknesses, but I crave corn. I actually eat it for dessert sometimes, but I know how it's not healthy for me at all. So I've talked about testosterone now, and I've talked about serotonin. And if we look at these hormones, neurochemicals, and so forth, it looks to be even a tiny bit more complicated. And some scientists have proposed this triple imbalance hypothesis, that violent behavior really depends on testosterone, serotonin, and cortisol, where we see higher levels of aggression when cortisol levels are low. So when I'm not showing a lot of anxiety or stress, testosterone levels are high, and then serotonin levels are low, as serotonin tends to inhibit those violent impulses. So if we have all of these things working together, that's when we're most likely to see violent behavior. And as I said, so we're talking about the amygdala, and we've talked about some attack behaviors, and now we're going to talk about fear and anxiety, and we're going to stay in the amygdala. And it's good to start out by explaining the Moreau reflex, as we're going to look at this startle response. The Moreau reflex when we're babies is, if you've seen the little Albert study, you've seen this. When we have a really loud noise, the baby's startle response is a Moreau reflex, where they kind of throw up their hands and arch their back and cry. As we get older, we don't throw up our hands and arch our back and cry, but we maintain a startle response. And I'm going to go ahead and tell my story I tell a few stories on myself here as we get into overactive amygdala. So when I was in graduate school, I took an eye movements and cognition class, and my professor was going to show us how when you have the loud clap, so I was on the eye tracker, that your pupils get larger, and that's part of our startle response. I jumped, so nobody saw my pupils get large. That it was a really intense startle response, and people have different intensities of their startle response. But what we typically see is we have this auditory information goes into the cochlear nucleus in the medulla, and some of that information gets sent directly to the pons. And if you remember, the pons when we're sleeping is stopping the muscles from moving. When we get a loud noise, the pons is going to command the neck muscles to tense. So if you can get somebody to stand behind you and kind of clap or do something when you're not really expecting it, you can, and you notice and think about it, you'll feel the tension in your neck. We don't learn to fear loud noises. This is something we are born with, but our moods and the current situation can modify our reactions. And I've already just discussed how there are individual differences in the way we react as well. The amygdala plays an important role in classical conditioning. So if you remember, I went through this really early on with my different brain regions when we talked about neuroanatomy. And if you put on the first exam that the amygdala, and you just put fear, I said that's not enough. There's more to this, as the amygdala is not just involved in fear and aggression and anger. The amygdala is involved with other emotions, and it is it's important, plays an important role in classical conditioning. And so all of this is really strongly intertwined, right? If I'm going to try and stick with, if I'm seeing a light and getting shocked, I'm being classically conditioned to where when I see that light, I'm going to have an anxiety response, even when I'm not shocked, right? I'm classically conditioned to have this negative response to seeing that light. If I'm going to hear a sound and I get food or get something sweet or something pleasant, I'm going to use food because that's what they do with animals. If I hear a sound and get food, pretty soon as I hear that sound, I'm going to have this relaxation response and probably some salivation and so forth as well, right? As this classical conditioning. And that kind of learning is taking place thanks to the amygdala. So we see an effect of classical conditioning on the startle response. Again, the startle response itself is not learned. That's something we're born with. But whether or not, so if I've been seeing a light and getting shocked, now if I see a light and I hear the loud noise, my startle response is going to be larger. If I've been hearing a sound and getting food, I'm going to hear that sound and then hear the loud noise, and my startle response is going to be lessened. So what they've found is that damage in the path from the amygdala to the hindbrain, that we still see a normal startle response. Again, the startle response is not learned. That's going to be something we have. But those signals of danger or safety, they don't modify the reflex in the same manner. So that's damage from the path from the amygdala to the hindbrain. And I want to go through some of these nuclei, these areas of the amygdala. So in the lateral and basolateral areas, it looks like that's where we are taking in that information, that visual and auditory information, so just that sensory information from the world. And then we're relaying that information to the central amygdala, and the central amygdala is putting that together with information from the thalamus coming in about pain and stress. And so as we learn to have a fear, we're going to strengthen the connections on this route from the lateral basolateral areas to the central amygdala. That's going to be strengthened with learning of fear. So it's those areas of the amygdala, and here's a nice picture that shows the amygdala, and then you can see how connected it is with the hippocampus. And you can also see this small band of fibers, the stria terminalis, and I'm going to now talk about the bed nucleus of the stria terminalis up there. As we get this long-term generalized emotional arousal, so if we are having a more generalized arousal or a generalization of fear responses, this appears to influence or be influenced by the bed nucleus of the stria terminalis. So people with a lot of activity there are going to have more generalized fear responses. We might talk about this as one example of this would be people who have PTSD, right? They're going to have a really overactive bed nucleus of the stria terminalis.